de una serie de charlas en la que estamos hablando sobre la relación entre los microorganismos acuáticos y la materia orgánica que encontramos también en el agua. Hoy tenemos el gusto de tener con nosotros a Masumi Stadler. Ella va a hacer una presentación de parte de los resultados de su trabajo de grado. Esperamos que sea de interés para todos ustedes. Y antes de empezar, muy brevemente quiero nuevamente, para quienes se han venido incorporando a esta serie de charlas, recordarles que estas presentaciones forman parte de un proyecto conjunto que estamos desarrollando entre el FRAPESPI de Brasil y la Universidad de Antioquia, en el marco de algunas ideas que hemos venido desarrollando en la red latinoamericana de ecología microbiana acuática, µSudAqua. Y bueno, podemos hacer estas transmisiones gracias a la ayuda de la Escuela de Microbiología de la Universidad de Antioquia y a la gran colaboración que hemos tenido de parte de la Red Colombiana de Limnología, que nos permite transmitir a través de su canal de YouTube y también depositar los videos. Por lo tanto, quienes no puedan estar el día de hoy, lo podrán ver más adelante. Em, Simplemente quisiera recordar antes de dar inicio a la presentación que la presentación estará transmitida tanto a través de Zoom como a través del canal de YouTube. Y Masumi va a hacer una presentación de cerca de unos 40 minutos. Les agradecemos que vayan haciendo sus preguntas a través del chat de YouTube o a través del chat de Zoom, en cualquier caso, y tendremos al final unos minutos para tratar de interactuar un poco y responder algunas de las inquietudes. Eh, Voy a cambiar ahora a inglés para presentar a Masumi. Masumi, hello, how are you? Welcome to this talk. Thank you very much for again for accepting being here. We are very pleased to have you here, and I think for the purposes of the µSudAqua network and for the people that is joining us through the YouTube channel of the Colombian Limnology Network, it's going to be very interesting to hear the ideas that you are going to share with us. Just for everyone, I would like to tell that, like to make a brief introduction of Masumi. Masumi started with a bachelor degree in the University of Vienna, and she started working with the concentration and the quality of DOC in leaf litter. And after that, she did a master studies at the University of Vienna again, working again with the DOC using fluorospectrophotometry. And more recently, she has been doing her PhD studies at the University of Quebec in Montreal, working with Paul Del Giorgio. And today, she's going to present us part of the results that she has been obtaining from the PhD studies. I know that Masumi, you already have a paper published with this information, but I understand that you will show us some other data that perhaps you are preparing for a new paper. So thank you very much for being here. And Masumi is going to be talking about following the flow of water, exploring microbial dynamics along the terrestrial-aquatic continuum. So Masumi, please, we will start with your presentation with about 40 minutes, and after that, we will have a few questions that people are going to be asking through the Zoom and YouTube chat. Thank you, Matsumi.
Perfect. Thank you so much for this lovely introduction, Juan Pablo. So as Juan Pablo said, I will be presenting some of my PhD work, mainly my first chapter that has been published already, but also adding some new data for my second chapter. So I completely forgot that you were going to do a short introduction for me. So I have a mini slide. I just wanted to mention that I did, DOM research in my master's where I focused more on fluorescence and absorbance. And I did the old school microscopy to enumerate microbes. And for my PhD, I dived into this high resolution, highly computational field. And it has been a challenge. I mean, everyone who has started with this knows how much information it is to get into this topic, but it has been also a very exciting one. So I'm very excited to share with you today what we have learned in my past few years. So let's start talking about microorganisms. I don't think I need a lot of introduction of microorganisms in this community here, but I just wanted to do a quick recap. This is a beautiful picture of an oral microbiome that might be currently living in your mouth. But within freshwaters, also microorganisms play a crucial role. They're decomposers, they consume matter that was essentially once alive, either of terrestrial or aquatic origin. And then microorganisms degrade and consume and continuously transform this dissolved organic matter that contributes essentially to this diverse pool of various compounds that we can find in the environment. And that can also contribute to emissions, essentially, because they are respiring what they're consuming. And deciphering kind of this interaction of DOM and microbes has been a decade or 2 decade long, study. But with high resolution, people were hoping that finally we can find more answers to the very inconclusive results that we have found previously. But I think one problem with high resolution is that we have kind of this computational and interpretational challenges that we face. So if we think about DOM assemblages and microbial communities, what the literature usually tries to do is link the bulk DOM with the bulk community. But recent evidence suggests that within both fractions, there is a part that is seemingly unreactive. So for DOM, there are persistent molecules, and for microbes, there are dead and dormant microorganisms. The persistent DOM pool somehow evades the degradation by microorganism. And when we try to link the two together, it might not be surprising that we don't find very conclusive results if there is a large fraction that is seemingly unreactive. So a major part of my thesis is to try to find these more reactive units within both assemblages. So for DOM, that's photo and bioreactive DOM compounds. And for microorganisms that are more active within certain locations, and then try to link the two to each other. Another facet of freshwater ecology, I think, and that is the connectivity of aquatic landscapes. We heard a very interesting talk from Michaela the other week, and this builds on this thing that, we should probably look not at individual ecosystems, but look at the whole network as a whole. Because when we think about it, we have microorganisms that enter the headwater streams through the terrestrial landscape or through the air, and then they're carried downstream as they kind of flow through different kind of ecosystems, like lakes and reservoirs, as well as rivers, as they move along the landscape. And the same happens essentially for DOM. There are analogous processes. We have terrestrial DOM that enters the aquatic network and then is carried downstream. Some of the DOM is degraded and decreases, while others might accumulate as it moves down the landscape. So we wanted to kind of use this connectivity as a tool to understand spatial behaviors and help us understand how those interactions lead to the microbial processes that we might find at certain points within this landscape. So the main kind of objective of my whole thesis was to truly understand microbial DOM interactions and how they relate to processes. We need to identify the reactive parts of both pools and consider their dynamics along a true hydrological continuum. So my thesis was kind of split into 3 chapters, where first I only looked at microbial community assembly along a hydrological continuum. And then we tried to link microbes in DOM in my 2nd chapter, and the 3rd chapter is on microbial processes. But for the sake of time, I will only present my first 2 chapters for today. The first one is published, so you can read it up later if you're interested. And the second one hopefully will be published in a near future. So let's dive into our study site. We're located in Canada. Our university is in Montreal. And the watershed that we study is in northeastern Quebec, around 13 hours away from Montreal. It is part of the Côte-Nord region. And this river, La Romaine, is around 475 kilometers long, and its watershed is around 14,000 square kilometers. I tried to find the equivalent sized river in South America, and I found that I think Rio Catatumbo, which is located between Colombia and Venezuela, is around the same length, but I think the watershed is a bit bigger. But just for you to get a grasp of the magnitude of the river that we sampled. And this La Romaine River has been a major, a long-term study of our group. So we have been going there to sample this watershed since 2015. It has been a very interesting case study since we were able to follow as the river was dammed with 3 reservoirs. Now in the end, there was also a 4th reservoir added, but it is not part of my PhD thesis anymore. And we continue to have the seasonal sampling and annual sampling where we cover the spring, the summer, and autumn. The spring in Canada is characterized by snowmelt and high hydrological influx into the river network, and the summer has a very higher temperature and lower hydrological connectivity. And we continue to sample DNA as well as RNA, starting in 2016. And then in 2018, we also added processes. So we have a very large data set of both DOM, DNA, RNA, over multiple years. And we tried to sample this watershed over multiple habitats, including soil, soil waters, headwater streams, the river, and of course the built reservoirs, to have this kind of entire watershed perspective for each of these projects. Now, before I dive into individual chapters, I wanted to acknowledge that the land on which the fieldwork was conducted is on the traditional and unceded territory of the Innu community. And the laboratory work was conducted on the land of the Kanien'kehá:ka people, as well as the Métis people, and I am immensely grateful to be working on their beautiful land. Now, for the first chapter, where we kind of focused on microbial community assembly, we wanted to have this network perspective, like I mentioned before. But one thing that we really wanted to explore was this idea of an upstream history. So since aquatic networks are majorly driven by a passive flow of water, and microbes do not need to actively swim around, they can just passively be carried with the water. There is this notion that any point within the network, the community that is present in that system should be influenced by what has happened upstream. So to put this into a kind of microbial assembly context, we have microbes entering the water, then they are carried downstream with the flow, And then you could enter a lake and the environmental conditions change, and some microbes die while others may not survive, may grow. And then whatever comes out from the assembly process that happens is then carried downstream. So there's this kind of history context that has been largely not explored within microbial community assembly. Just to quickly recap some assembly processes, I think many of you are familiar with these, so I will just quickly go through them. But we have, in theoretical ecology, there are 4 major assembly processes that are defined. We have selection, which essentially means that microbes that are adapted to a certain environment are favored in certain conditions and grow. Then we have dispersal, which can be somehow affected by a species trait, since if you have the ability to swim against the current, that might affect the dispersion of that microbe. But there is also this passive physical dispersion process, which is also often referred to as mass effects. Then we also have processes that are more on an evolutionary timescale, which are diversification, which is mutation and the creation of new species, and drift, which is stochastic events and population changes through extreme events, for example. And these 4 processes are on a scale from being random to deterministic, meaning being dependent on the species trait. In our particular example, we are more interested on ecological timescale processes that are selection and dispersion. So again, to kind of put this into our context, if we have a lake within the network and there is a stream flowing into the lake, we have microbes that are passively dispersed into the lake. So we have a mass effect flowing into the lake. And then as the conditions change in the lake, some microbes might grow while others might die that are more adapted to the environmental conditions, which is the selection process. And whatever comes out of the lake is a balance between the two. So one of our main questions was, well, how does this balance between mass effects and selection change as we go along the continuum? And we wanted to explore this importance of the upstream history. The 3 major questions we asked were, well, how does microbial community assembly change along a true terrestrial-aquatic continuum? How is it affected by seasonality and hydrology? And who along the rank abundance curve is responsible for these community shifts? To quickly go through a microbial pipeline, so usually you have environmental samples, you do an extraction process for DNA and RNA. For RNA, you have one more step to transcribe cDNA from RNA. And then the downstream process is all the same. You amplify the sequences, you sequence them. And we focus particularly on the 16S RNA marker, which is a taxonomic marker. And then you do some bioinformatic magic to achieve some degree of abundance measure. One question is, of course, how to represent microbes. Traditionally, people have used the 70% similarity threshold into operational taxonomic units, which may cluster microbes that have different functions together. So something that has been very popular in recent years is the Data2 pipeline that we also used, which gives you 100% similarity threshold, and they're called amplicon sequence variants. Now, for our particular exercise, since we had DNA and RNA, we wanted to be able to match the 2 together. So since RNA is a little bit more volatile and it degrades quicker, it is expected that you cannot find the same DNA and RNA with 100% similarity threshold very often. And there also has been recent evidence that when a species has multiple copies of the 16S RNA gene, the sequence can be not 100% identical. So you might end up with multiple ASVs for the same species. So as a compromise, we ended up using a 99% similarity threshold to be able to cluster species together and also be able to match the DNA and RNA as best as possible. And then what we usually do are multivariate analyses to look at the taxonomic shifts in the composition. And this is a principal coordinates analysis with Bray-Curtis dissimilarity. And you can see here on the first axis, sorry, I forgot to mention that the colors are the different ecosystem types. So we have soil, sediments, streams, lakes, the river, the reservoir, and then we also have two separate sections, the upriver and the downriver, which is essentially the river before the reservoir cascade and then after the reservoir cascade. And then we have the 3 seasons in different shapes. And then the polygons represent the range of DNA and RNA within the multivariate space. And I think here you can see nicely on the first axis, we have the separation from terrestrial to aquatic ecosystems. So the communities diverge as soon as you enter the aquatic system. And on the y-axis, on the second principal component, you have a segregation of DNA and RNA. So this indicates that as soon as communities enter aquatic systems, the DNA and RNA pool starts to diverge a little bit, while in terrestrial influence systems like soils, soil waters, and streams, they're rather similar. And to add a third axis to this, we have a seasonal component within the aquatic samples where we can clearly see that there is a segregation. Let me see if I can put a laser pointer. You have the spring samples over here, and then all the summer, autumn samples that are diverging once you enter the aquatic system. Now, to come back to our question of how does the balance between selection and mass effects change, we try to come up with a new way to analyze this. So let me walk you through the method that we applied. So the PICO that we just saw was based on a Bray-Curtis dissimilarity metric, which is essentially an abundance indicator. So if you have an OTU matrix that you can see here, you have the individual samples and the different OTUs. And then each of these columns is the number of reads you can find for each of these in each sample. We focused on the DNA and RNA within the same sample. So the same sample, but their DNA and RNA. And we wanted to calculate how much they differ in the multivariate space. So what we calculated is this distance within multivariate space between DNA and RNA of the same sample for the abundance space dissimilarity, which kind of implies that whenever there is more reads in RNA compared to DNA, for example, that is an indicator that that OTU grew specifically in that system. That implies that there is some kind of selection going on. And then we also transformed this initial abundance matrix into an incidence-based matrix, which is essentially abundance and presence. And with that, there's a similar dissimilarity matrix called the Sorensen dissimilarity index that you can calculate with this. And this is more representative of what different taxa are between two samples. So with this, if we calculate the distance between the DNA and RNA of the same sample, if they're very far from each other, that means that they have very different taxa, which implies that if there are a lot more taxa in DNA than RNA, there's a lot of microbes that are kind of washed in that are not active within that system. So that is an indicator of mass effects. So by using these two metrics, we were able to kind of define that the Sorensen-based, incidence-based indicator will tell us more when there are more mass effects. And when the Bray-Curtis-based, so abundance-based index is higher, there's a stronger selection. These hypotheses of these two metrics were tested with simulations, and they mostly follow what we think they are doing. But I will not present the simulations today. If you have questions, I'm happy to show them to you. And then as a further step to kind of quantify the balance between the two processes, we calculated this delta distance by subtracting one from the other. And that means that when delta distance is very high, it indicates that many taxa are in common between DNA and RNA, but there are more numerical differences that make them move apart, which probably indicates selection. On the other hand, when delta distance is low, there are many different taxa between DNA and RNA, which probably indicates mass effects. So when we apply this to our terrestrial hydrological network, it's a little bit complicated plot, so let me walk you through it. So you have the, first of all, the x-axis represents the continuum. The y-axis is the distance within the multivariate space. The colored points are the abundance-based distances, and the hollow points are the incidence-based distances. And then the three panels are the three different seasons. So if you look at this, we can clearly see that, first of all, the incidence-based distance is much higher in spring compared to summer, probably indicating that there are a lot of not active bacteria that are flushed in spring, pushing the entire network higher than in summer, where there is more, when there are more DNA and RNA common. So probably indicating that there are less mass effects. When we look at the abundance-based incidence, we can see that in general, they're pretty similar with the exception of the soil sample where we have a big difference between DNA and RNA. But that is also reflected within the incidence-based distance. And finally, to ask the question, well, how does the balance shift? We can look at the delta distance that we calculated. So it seems that in spring, again, the whole baseline is much lower. So it seems that overall, mass effects are very strong. And the way I try to interpret the delta distance is more, rather than the numerical value, it's more the shift from one habitat to the other. So you can see here that first we have very low delta distances, probably indicating a lot of mass effects, but that increases once you go into streams. So there's probably some degree of selection happening in streams as well as reservoirs because we have a positive shift. When there's a negative shift, it probably indicates that there is more mass effects happening afterwards. So for example, after the reservoir, once we enter the river, we have a bit more mass effects, probably because, you know, a reservoir kind of shunts these microbes that are adapted to more lake-like environments into a river-like system. So they might become inactive in the river system since they're not adapted to these kind of conditions. And in summer, we can see that this is very low, but then we have this very sharp increase in the delta distance, which probably indicates this strong selection that goes on as soon as we enter the aquatic network. So with this, we were kind of able to find this balance between mass effect and selection along our continuum. And before I kind of summarize this chapter, I wanted to just quickly highlight some results from this very complicated plot, which here on the top left, you have essentially, again, the terrestrial aquatic continuum on the x-axis, and then the proportion of reads in the local DNA pool. So these are all the taxa that are in the DNA pool, and these are colored by where they were first detected in. So first in soils or soil water, streams, and so forth. What I want you to see here is that as we move down the continuum, we can see addition of different colors, indicating that we have addition of new taxa into this non-reactive DNA pool, since these are all the taxa that do not have any RNA. This probably implies that we have this kind of upstream history, that more and more taxa are loaded into the hydrological network as we move downstream. However, when we look at the most reactive taxa within the RNA pool, we have this clear dominance of soil and soil water derived microorganisms that seem to be the most reactive across the entire hydrological continuum, with the exception of the estuary. So in the estuary, we have a few taxa that become very active that were first detected in the estuary, which makes sense because it's a completely different environment. So to kind of sum up these findings, so we kind of found that mass effects seem to be present at all time. I think in the literature, we often read it's only selection or it's only mass effects. But I think we have to keep in mind that these two processes are always present. And it's always a balance between the two. And it seems that in general, spring is dominated by mass effects and summer is more dominated by selection. However, among the seasons where within the hydrological network, the strongest selection is occurring shifts. So at high flow, we have more selection in longer residence time systems like reservoirs or lakes. And at low flow, that kind of intense selection shifts upwards in the network into rivers, where we have many more reactive selection happening there. The only exception to this kind of overall patterns were streams. They always had a very high mass effect, but also seem to be sites of strong selection, as we always saw that positive increase in our delta distances, which is quite exciting. And I always believe that that might be linked to biofilm because, you know, it's a very different environment and it's kind of stable across the two seasons. So a few take-home messages from this first chapter is that the first two I already summarized in the first slide. So to just add a few results and conclusions that I wasn't able to show today, but that we found in our results was that the selection itself actually occurs across the entire rank abundance curve. It is often assumed that only the most abundant taxa are contributing to local processes, but we were able to see that even rare taxa were actually positively selected for in certain ecosystems. And we also found this very strong historical imprint within the dead and dormant DNA-based community that I pointed out earlier in the results. All right, so that was my first chapter. And before we go into my next chapter, I have a quick water break. So in my second chapter, I tried to kind of link microbes in DOM. This is still ongoing work. I do not have it all figured out, so please bear with me. But so the whole idea is that links back to the introduction that I had earlier, is that we wanted to kind of extract the most reactive parts within both microbes and DOM and try to link the two. One major issue here when we talk about connectivity is the problem of distinguishing mere hydrological correlation that we might find from actual causal interactions. So that is something that is extremely complicated when in terms of DOM and microbial community dynamics. And I don't think I will have the answer at the end of this chapter, but I think it is a first step forward in this kind of interrelated, complicated relationship. So the three questions that we tried to answer in this chapter was, well, what is the proportion of unreactive versus reactive microbes and DOM? Do chemical and phylogenetic similarity underlie spatial patterns? And can we distinguish mere hydrological patterns from causal interactions? I actually had forgotten to mention in this slide before that we wanted to use the connectivity as a tool to identify reactive moieties in both assemblages, because the problem of DOM in particular is that we don't have a counterpart like RNA. that we have for microbes. For DNA and RNA, we can say, okay, if they're in RNA, they're more active and they're more reactive to the environment. But for DOM, there is no such thing. So the way that we thought of tackling this is if you have a spatial gradient, it could be any gradient. We will use hydrology as our gradient, but you can have a pH gradient or a fertilizer gradient or anything. And then you can try and look if a DOM or if a microbial unit changes to that environment. Because if it was unreactive to the environment, it should be stable across systems. So we will try to use this kind of spatial behavior as an inference for reactivity for both assemblages. So similar to the microbial pipeline, we have a DOM pipeline. It's actually quite similar. So you have samples, you do an extraction, and then you have an extract, and then you analyze it with this mega magnet, and then you do some mathematical transformation to have peaks, which is a mass spectrometer, and then you assign molecular formula to these peaks. You might ask, well, what is a molecular formula? It is essentially the elemental composition of a compound. So here you would have, this is a molecular formula. And all of these 5 compounds that you can see here have the exact same molecular formula, but they have different structures. So that's one caveat of FTI-CRMS, is that it is not able to distinguish different structures. And the gradient that we used to kind of do this spatial behavior analysis was flow-weighted travel time. For the sake of time, I will not go into much detail here, but essentially it is a metric to capture how long a parcel of water has been traveling within the landscape. And traditionally, this is only been done for streams. So whenever you have a stream network, this is a very GIS-based calculation. So that's why most people just say that, okay, in this network, there are no lakes. We will only focus on the streams and rivers and calculate how much it takes to pass through the landscape. But we try to incorporate different habitats into this metric by calculating the velocity within the network, depending on the ecosystems that we have. So we modeled a velocity using catchment area across the whole network. Then we also modeled discharge using catchment area across the whole network. And then whenever the network was covered by a lake, so when there is a stream that is actually flowing through a lake or a reservoir, we assigned those stream reaches that were under lakes and reservoirs the velocity that we calculated for lakes and reservoirs, and that we derived from the water residence time that we calculated. And we then sum this up along the network. So as you move down the stream, you sum up the water residence time within each pixel, and then you end up with the travel time, and then you weigh it by the discharge whenever you have a confluence merging. And this ends up with something like this. So this is only the main channel highlighted with the travel time that we calculated. In essence, whenever you exit a lake, you have a very high travel time because it slows everything really down. But once you exit the lake, as there are new streams flowing into the river, the travel time actually starts to decrease until it hits another high water residence time system where the travel time then increases again. So it is essentially kind of a water residence time metric, but in a more network context. And then we use this travel time gradient to model individual spatial patterns of microbes and molecular formula. We had this kind of decision tree that fits different models for each individual spatial behavior and decides, well, for this microbe, a linear model fits the best, or for this one, a non-linear model fits the best. And this allows for non-linearity, which is often neglected in these kind of community studies. And then using a few model coefficients and where peaks are located within the spatial gradient, we were able to define the spatial behaviors into 5 major categories. So whenever a model was not significant, they were deemed to be unreactive. And then we have some microbes and molecular formulae that increase along the spatial gradient that might indicate some degree of production, a constant influx or accumulation, and that can be linear or nonlinear. And then we have the counterpart of some microbes and DOM that decrease along this continuum, which might indicate some consumption, death, or dilution. And then we also have some microbes and molecular formulae that have more like a local peak. So we have a unimodal shape that we also allowed for this. So all of these colored spatial patterns were then deemed to be reactive to the environment, and everything else was non-reactive. And if we then proportion how many of the models came out reactive versus unreactive, we could see that overall, both proportions, DOM and microbial, was largely dominated by unreactive moieties. For microbes, we only found around 15% that were reactive versus DOM that is around 44% reactive. And then we had the spatial patterns, and it seemed that across the campaigns, the microbes were quite dominated and consistently dominated by increasing microbes, versus the DOM had a more interannual pattern rather than seasonal, where we have more decreasers in 2015 and more increasers in 2016. And we are speculating that this might be because there was a freshly flooded reservoir between those years. Now, the next question was, well, is there any similarity between these spatial between the microbes and OTUs that fall in the same spatial pattern in terms of phylogeny and chemical similarity? To do some kind of similar phylogenetic exercise with DOM, I calculated a tree by hierarchical clustering. So this is I think it's a unsupervised or semi-supervised machine learning approach, where I essentially gave the model the different kind of chemical components of a molecular formula, its mass, its elemental composition, its aromaticity, and then it clustered them by their similarity. And we could find 5 different clusters that were different in their aromaticity, in their degree of nominal oxidation state of carbon, and so forth. So once we plot this with the spatial patterns here, so here on the left, you have the DOM tree, and then on the right, you have the microbial tree. You can see that, first of all, the microbes are very empty. So we have much less reactive OTUs within microbes and many more within DOM. But if you look between the rings here, so these are the different campaigns, we can see that for some microbes, it seems to be always the same pattern. So this one in particular, for example, is always increasing along the continuum. Whereas for DOM, there's kind of a shift between the campaigns. So to actually test this, we calculated this metric that I will not go into detail, but it essentially calculates whether things are spread across the tree or rather clustered across the tree. And it seems that for microbes, there is a strong clustering of those that are increasing along the spatial pattern. So they're more similar to each other, versus those that die along the continuum or disappear are more dispersed across the whole tree, meaning that it's probably, you know, not a, how do you say? Well, it's not a phylogenetic trait to be declining across the hydrological continuum. And we wanted to see if we can find some functional underpinning to this as well. So we calculated a 16S copy number, but there was no difference among the spatial patterns. And for DOM, we did the same exercise across the hierarchical tree, and it seems that they're all quite clustered across the tree. However, there was no major difference between increasers and decreasers. It was only that the increases are more clustered than the unimodal patterns, but there was an extremely strong difference between in their functions. So we had, in terms of aromaticity and this nominal oxidation state of carbon, which is an indicator of how highly favorable a compound is when a microbe degrades it. So we can see that there is some kind of chemical similarity as well as phylogenetic underpinning to these spatial patterns. And I would like to wrap up by showing you this kind of imaginary exercise that we did. So as I had mentioned earlier, there is always this correlation and causational problem that we have. I will not go into all the details, but the next step is, of course, is to link the two to each other. So we would correlate individual spatial patterns of OTU and DOM. And you can imagine, for example, if here, this is the easiest example. If a microbe increases along the hydrological continuum and a molecular formula decreases along the continuum, in a causational context, this might incline that the microbes are consuming the DOM and therefore they're growing. But in a correlational perspective, it might be simply unrelated sources. This DOM might be of allochthonous origin, while the microbe is of autochthonous origin. So you might find these correlations that are have no causational underpinning. And just to give you a sneak peek into this, so we tried to calculate many different things. We tried a lot of things, but I think what we were able to see is that when you look here, so here are all the 4 different relationships, and the line here is the average across all clusters for that specific group. And if it was simply correlation, I would say that each cluster should have roughly the same correlation coefficient, but they're not. So that implies that some clusters are more related in specific relational patterns than others. And that is something that I still have to sit down and really digest everything that we see here, because it is also not seasonally consistent. So it's very hard to decipher everything. Yeah, I just wanted to highlight this one where you can see clearly here. This is the one where the microbe would grow, and then we have DOM that is decreasing, and we can see here that cluster 1 is a very highly aromatic and highly energy favorable compound group, and these are the most related after this one. So, this is exciting stuff that I'm still trying to digest, but hopefully at one point I can present to you. Okay, to summarize the second chapter, so it seems that we have a really large fraction of unreactive microbes and DOM that we have been traditionally trying to link to each other. And within the significant patterns, we have more increasing microbes along the hydrological continuum, whereas DOM had this kind of interannual pattern with no clear seasonality. And spatial patterns are influenced by phylogeny and chemical similarity. And even after doing all of these exercises, it is still very difficult to distinguish correlation from causal processes. And these are, I think, things that we should keep on exploring to figure out how they're related to each other. With this, I would like to thank all the members of our laboratory and everyone who has been going to these campaigns. These are major, large campaigns that no single person can do alone. So I would just like to highlight a few special people that are very, very crucial to this La Romaine project. And of course, I would like to thank you for inviting me today and listening to me today. And if you have any questions, even after this talk later on, I'm happy to answer any questions by email or any other platform that you'd like to contact me. Thank you.
Thank you, Masumi. Great work, very interesting. There are lots of ideas. I forgot to mention at the beginning that one of the reasons why we decided to invite you is because we are involved in a project where we are trying to link DOM and microbes composition along different catchments in South America. So I found that that your presentation is quite inspiring. I think that's one of the ideas that we had with your presentation and the other presentations before, and the one that we will have in about a month with James Stegen. And I think we need a little inspiration based on your experience and other people's experience. So thank you very much. It was very clear, although it was complex. So I think we also need to digest a lot of information. But of course, about the first chapter, it's very nice. I think the approach that you applied, it's quite different to most of the approaches that the people are using in the literature. So I think we will have to discuss a lot, and perhaps it would be interesting to talk in a different environment with you and clarify so many things, because the metrics are seem to be complex. And actually, one of the things that I was thinking was, how do you think people are going to start using these metrics, or do you think it's going to be just the exercise that you are doing to show these patterns? What do you think if people is going to use this approach?
Right. So the whole idea of using a different metric than what is commonly used in the literature was that, and the metric that is usually used is based on the phylogenetic similarity that I had mentioned earlier. So the idea is that phylogenetically similar microbes should react similarly in certain environments, and that means there is selection. And we just wanted a metric that is simply based on numbers, without the assumption of, microbial traits and so forth. And I remember that James Stegen had also reached out, because I think one of the interesting things would be to actually compare the two metrics to each other. And maybe that is something that you could apply. I understand that the metric we introduced is very complicated, but the scripts in R are publicly available. So that is something that either if you reach out to me, of course, I can help you with that. And also, if you find the data online, the scripts are available. And everybody is familiar with, well, not everybody, but most of the people are familiar with multivariate analyses. And because that is the first thing most people do with microbial community composition. So I don't think that is the hurdle. It is more diving deeper into extracting the numbers and trying to link them to each other. But I think once you grasp the concept and how to do it, it is not as complicated as it looks like. I know that it sounds like false promises, but
yes, I think you're right that it takes a time, or people is just gonna use it maybe without thinking too much of what is happening in the script or whatever. But I also found very interesting this idea of, I don't know, matching the metrics for DOC and bacteria, and to test if they are changing. Very interesting. I'm really curious to see the final results of the second chapter. Hopefully you get there to the publication really soon.
I hope so.
I think we will be happy to see the final result, but the idea is really great. And also the conceptual framework that you presented at the end, it would be interesting to analyze it with more time. But I think the idea is brilliant. So hopefully we will see your paper soon around.
I hope so too. Fingers crossed.
So thank you, Masumi, again. There are a few messages. Congratulations, super interesting talk. Thank you very much. So I think at the end of October we will have the µSudAqua meeting, and for sure we will be discussing about your ideas and your conceptual framework, and hopefully we can meet and at some other time. Thank you
That'd be
very
great.
much for being here, and let's see how people react to the YouTube
Right.
talk that is going
I
to be around.
I just think there's a question. There is a hand
Okay.
raised.
Can you see it in the chat right now?
Yes. Rocío?
Well, no.
Alcantara. La señora Rocío Alcantara va a hablar.
Rocío, adelante.
Thank you. Thank you very much, Juan Pablo. Yeah, thank you very much for the nice talk, Natsumi. I'm not an expert in rivers and streams, so this question comes from my total ignorance of the system. And you mentioned as a conclusion that microorganisms are dominated by an unreactive part, as are mostly unreactive, meaning they do not contribute significantly to the biochemical processes, I guess. So I think this is done in water. Sorry, I arrived later, well, late in the talk. So I guess this is in the aquatic part of the streams, isn't it? It's a planktonic community.
Yes.
Oh, yeah. My question is, what's the role of the sediment communities here? Are they also contributing to the most of the biochemical processes, or they are also in a lesser way attenuated in this activity given the time of residence of the water? I mean.
Yeah, that's a very important point. I completely agree with you. I think it depends really on the ecosystem because, aquatic networks are so diverse. So within streams, you don't really have a sediment, but you have a biofilm community that really contributes a lot to the processes that are happening. And within more stagnant waters like lakes and reservoirs, of course, you have a big sediment proportion. Unfortunately for our study, we did not kind of include these metrics because it was not part of the whole sampling scheme from the beginning. But there are some studies that are coming out that actually contrast biofilm and sediments and how they are differently shaped. And I think a recent study was just published this year looking at this, and they found that sediments and biofilm communities are much more similar across watersheds and ecosystem ranges, and from the top of my head, I don't remember the fraction of reactive portions, but I do remember that they were very differently shaped and contribute majorly to the processes as well. So that is something that I think future studies should definitely look into.
Thank you very much. That totally answers my question. Thank you very much. Very interesting work.
Thank you so much.
Thank you, Rocio, for your question. And well, I think it has been a very nice talk. Thank you, Masumi, again. And
thank you so much.
I hope we'll meet together very soon.
That'd be wonderful. Thank you so much for having me again. It was a pleasure.
Ok, gracias a todos quienes asistieron a través de los canales de Zoom y de YouTube. Nos vemos en la próxima charla donde James Tegen nos va a hablar un poco de cómo utilizar estas metodologías que Masumi presentó el día de hoy, cómo obtener información a partir de estas metodologías, seguramente aproximaciones distintas a la que Masumi presentó hoy. Gracias y bueno, seguimos adelante. Gracias nuevamente a la Red Colombiana de Limnología por facilitar la transmisión de estas charlas y nos vemos muy pronto. Bye to everyone.
Bye. Thank you so much.
Thank you.
Okay, Masumi, thank you very much. Great talk.
Thank you so much.
It's really interesting what you are trying to do on your second chapter.
It
Very,
has
very-
been a long-term struggle.
So can you see the end or not?
Yeah, I'm supposed to submit my thesis next month, so I have
Okay.
to really squeeze out as much as I can. But no, I hope